Hello, everyone, and welcome. Welcome back to the last lecture on the innate immune system. And now we're going to talk about the interferon system. So far we talked about the complement system, and we talked about different phagocytes in our innate immune system, which we mainly focused on the macrophage and the neutrophil. And mind you, in the last video, we did talk about other granulocytes like eosinophil and basophil. They're not phagocytic, depends on who you ask, but the neutrophil definitely is, and then definitely the macrophage. And then now we're going to talk about the last part of the innate immune system, and that's the interferon system. So interferon system is an antiviral system. It's a very non-specific system. If this system is in play, cells in general become more ready to fight against the virus, or rather apoptosis, so the virus does not spread further. So how does this work? Again, viruses are not living organisms. Right now we have this whole coronavirus that is not a living organism. It's a virus, which is just a big swirl of genetic material with a couple proteins tacked on that seeks to replicate itself just by floating around. But yeah. So how does interferon system get activated? And then, mind you, interferon system is specifically for viruses. A pattern recognition receptor, which are like those evolutionary receptors we spoke about from before, who are very good at picking up red flag that we've evolutionarily learned that tend to be dangerous. So this happens. The macrophage becomes hyperactivated, and it becomes a IL-1 and TNF factory. It becomes more phagocytic. It grows in size, but it also is going to fire off warning signals to nearby cells. And this warning signal come in the form of type 1 interferons, which are interferons alpha and beta. These interferons bind to interferon receptors on cell throughout the body, so like endothelium, hepatocytes, you name it, right? And then these cells pick up they get the signal via this interferon receptor. What does this binding do? This binding sets off an intracellular cascade that essentially allows all cells or pertinent cells in the body to produce internal antiviral proteins in the background. Basically, it's the macrophage telling the nearby cells, hey, I just picked up on a red flag. We have this shady virus in the area. Here's a warning signal. Just be on the lookout. And so the cells will just keep doing what they're doing. They form their normal metabolism. In the background, they start to synthesize a little bit of viral proteins on the side. And this way they're prepped, antiviral proteins on the side. And this way they're prepped should that nearby virus from that nearby signal find its way into that specific cell. Quite amazing, really. And then however, there is also another type of cell that's very good at the type 1 interferon production, and that's going to be the plasmacytoid dendritic cell. These have nothing to do with the normal dendritic cell that we talk about, and you've probably heard of, and we're going to talk about later, the antigen presenting one. This is nothing to do with it. So just this is called dendritic because of the name, but it has nothing to do with it. Plasmacytoid dendritic cell. Actually, if I'm mistaken, even some dendritic cells don't even have dendritic appearance. I think this is one of them. I think this one's actually just like round, like most cells. That's not the point. So plasmacytoid dendritic cells are type 1 interferon factories. So basically what they do is that they gulp up their surroundings periodically. They have a ton of toll-like receptors inside their bodies, or cytoplasm rather. And we said TLR7 viral DNA, TLR9 viral, sorry, it's the other way. It's aside the point. It's actually bacterial, but sorry, no, I had it right. TLR7 is viral RNA, and TLR7 is viral DNA, right? I had it right the first time. And because of all these toll-like receptors, it's very good at picking up red flags. The second it picks up a red flag, it can just gush out a ton of interferon alpha and beta, and it's going to flood the body with it, and it puts big portions of the body on guard in case of a virus shows up into that cell, the cell's already primed. And should the virus enter the cell, that cell will have some antiviral machination proteins already in place, and also it'll be ready to apoptose, essentially kill itself in a controlled fashion to, take one for the team, not allow further spread of the virus. Because remember, the more cell the virus gets inside, the more machinery it has to its disposal to replicate. These are my favorite leukocytes, called the natural killer cell. They're actually also lymphocytes. They're related to B cells and T cells, more closely T cells, but they are not adaptive immune system. They are innate. Really, what they are, they're a bit of a mix of a neutrophil and cytotoxic T cell, but this analogy doesn't do anything for you because you need to know the specifics, or you want to know the specifics. I don't know why you're here. Thank you for being here, but I don't know why. They are short-lived. They live only for about a week, and then they extravasate from the bloodstream into the tissue, just like the neutrophils via that mechanism we discussed with the SLIG binding L-selectin and then the integrin binding ICAM on the endothelium. Natural killer cells secrete interferon gamma. They can basically help T cells to wake up macrophages in this regard. But mind you, even though they're a bit like T cell, they don't have T cell receptors. They have their own receptors. And they actually look if MHC is missing. And so we'll talk about that later. But rather than looking for specific antigens, natural killer cells tend to look for if a cell is missing an MHC molecule, that must mean something is funky is up with the cell, and therefore command the cell to die. But yeah, NK cells are natural killer cells are very rich in toll-like receptors. And so they are very good at picking up those evolutionarily designated red flags. They're sensitive to and can be activated by interferon alpha and beta, so they are put on guard by interferon alpha and beta. And again, they give the cell the suicide signal should something be up with that cell, whether it picks up something with its TLR or if it picks up that cell is not projecting an MHC molecule, MHC1 rather. So what's the mechanism of NK cells killing? Well, the NK cell uses a surface protein called perforin, and that's what it does. It perforates, so it can perforate other cells, and then in that hole that it gets perforated, the perforation it just made with perforin, it'll dunk granzyme, specifically granzyme B, I believe, inside the cell, and granzyme is highly toxic to cells. So it pokes a hole, then pours poison inside. That's what NK cells do. Also, NK cells also use a surface protein called Fos ligand. Throughout the body, we have a surface marker on like our cells, endothelium, nephron. The cell that line the nephron and the kidney tubules. What else? Arbitrary hepatocytes, intestinal, endothelium, epithelium. Any cells, right? It's going to have something called Fos, and Fos is basically a suicide button, but not everyone has access to that button. Cells that have Fos ligand have access to that suicide button, and so I think NK cells, and then I think some T cells have it as well. So if Fos is pressed, the cell will die, and so that's another mechanism in which NK cells can kill if they know something fishy is going on with that cell. And so that's what I was saying about MHC class 1. If the NK cell does not see MHC class 1 on cell surface, it will kill the cell. However, if MHC1 is present, NK cell won't kill it. So it's a very cutthroat type cell. You either have MHC1 or you don't. You don't have MHC1, you die. Mind you, mainly inspects the nucleated cells, right? So you don't really have your NK cells going around killing red blood cells who are anucleated. It's just if there is supposed to be MHC1 there and it's not, NK cell will kill you. That's what it is, right? So that's what that is. So there's some interplay between NK cells and macrophages, and this is like the last part of this lecture. Mind you that the NK cell can detect the red flag with its toll-like receptors, and it can release interferon gamma to wake up the macrophage to put it into a primed state. At the same time, remember that the macrophage has toll-like receptors as well, and it can pick up on red flags as well. It can pick up on the same red flags, because remember, we had like the same 4 and 9 for RNA and DNA for the virus, right? However, but mind you, if when this happens in the macrophage, it enters the hyperactivated stage, and it's going to produce IL-1, which is the pyrogen tumor necrosis factor, which performs killing against cells and through the avascular means in case of cancer, but also something called IL-12. And I have a mnemonic here for where IL-12 comes from, but IL-12 is essentially hyperactivates NK cells, right? So when the NK cells even more activated, it produces even more interferon gamma, which wakes up even more macrophages. So it forms like this big loop, this big interplay. Also, mind you, um, uh, the NK cells will, upon hyperactivation, not only do they produce interferon gamma, they also produce something called interleukin 2. Interleukin 2 is a natural killer cell growth factor. So in a sense, upon hyperactivation, natural killer cells can self-stimulate to proliferate. That's one way to put it, right? And so that's all for the innate immune system. Thank you for sticking with us if you stuck the whole time. We talked about a lot. But with this basis, I really do hope you have better understanding of the innate immune system and can appreciate it. Or maybe you're not here to appreciate anything; you're just here to pass your exam. Whatever reason that you're here, I hope you found what you're looking for. But thank you, everyone. I really appreciate you being here, and I hope you're enjoying this series. If you'd like to tell me what you think or you request something, please let me know. We have a lot more content to come, and these are my sources. All right, everybody, take care. Have a good day.